Good evening, everyone, and welcome to the 2025 Allan and Maria Myers Lecture. I'm Peter Van Weingarten, CEO of the Florey Institute of Neuroscience and Mental Health, and it's my great pleasure to welcome you this evening. I'm so pleased to see all of you here, and we're also delighted to be joined online with a large number of people watching the lecture remotely. So I'd like to begin by acknowledging the traditional owners of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present, and I extend that respect to any Aboriginal and Torres Strait Island people with us this evening, either in person or online. By way of housekeeping, the exits are at the front and at the rear, and the toilets are at the top end of the foyer. Please note that we are live streaming the lecture tonight, and that will be recorded. So the Allan and Maria Myers Lecture is the annual keynote lecture of the Florey Institute. Each year we invite an international leader at the pinnacle of neuroscience research to share their work with us, and I'm delighted that Professor Marcus Conrad will speak to us tonight. I extend my sincere thanks to Allan and Maria Myers, whose generosity and visionary commitment to scholarly exchange has made this evening possible. It's now my great pleasure to invite Professor Scott Ayton, Head of Dementia Research at the Florey, to introduce our speaker. Welcome, Scott.
Thank you, Peter, and it is my great honour to give this introduction of Professor Marcus Conrad. Marcus is going to be talking about something called ferroptosis, and this was actually coined by another colleague of ours, Brent Stockwell, back in 2012. But Marcus is regarded as the discoverer of this cell death modality, having independently identified this pathway, but he didn't assign it a name back in 2009. Now, I've got a few slides here to say why we think ferroptosis is such a big deal. We all know that cell death is one of the major areas of biology that has preoccupied research for both cancer and degenerative diseases and ischemic diseases as well. And you can see the cell death, the publications in cell death, and have predominated by apoptosis, which of course we all well know. In the last few years, we've had a little bit of a dip because there's been this emergence of other cell death pathways apart from apoptosis. And even still, people refer to apoptosis when they're calling all forms of cell death. But now we understand there are different types of cell death. And if we look at this other, we can look at one of the first new identified cell death pathways called necroptosis. And you can see there is an emergence and progressive increase of publications. Next came pyroptosis, and you can see that's even got some more momentum. Then a little bit later came parthenotos, and that didn't really get a whole lot of momentum, did it? It's still very important. But what I think is really striking is the trajectory of ferroptosis papers, particularly in recent years. This is a field that is exploding, and it's really Marcus who I think, in work that began in his thesis dissertation and continued in the 20 years since, Marcus has identified all of the major regulators of this pathway, and he's going to talk about them today. GPX4, FSP1, ACSL4, SLC-A11. These are fundamental proteins and genes involved in ferroptosis, and Marcus is the discoverer or co-discoverer of all of them. In addition, he has developed the industry-leading small molecule inhibitors and inducers of ferroptosis. And he is published with rapidity in journals such as Nature and Cell. Marcus is ranked #1 in ferroptosis by ExpertScape. And prior to this year, he acted as the convener of every major ferroptosis meeting globally. So he really has played an outstanding role in shepherding this emergent field from the discovery of fundamental orchestration of the cell ferroptosis induction and defense through now to this pivotal time of clinical translation. So Marcus is the research director of the Institute of Metabolism and Cell Death at Helmholtz Munich, and he's also chair of translational redox biology at the Technical University of Munich. He's authored over 180 scientific publications, has been recognised as an ISI highly cited researcher since 2020, and he's received numerous prizes and awards, including some very significant ones that I'll mention: the Paul Martini Prize, the Glenys von Pergamon Prize, and the European Cell Death Organisation Honorary Award. Marcus is a true giant on the field, and we are grateful to Allan and Maria Myers for providing this opportunity. Would you join me in welcoming Marcus to the stage?
Wow, what an introduction. It's the first time I hear all these things. And yeah, thanks, Scott, for this very wonderful introduction. Also, of course, thanks, Peter and Florey, and of course, Allan Myers, who I had the pleasure to meet him yesterday for a wonderful lecture at the Newman College. And now I'm here and present similar stuff, of course, because I think this is a Florey, it's a neurodegeneration center. And I skipped most of the cancer slides, so if you want to listen to them, there will be another event occasion tomorrow, but I think Scott will talk about it a little bit in a minute. Okay, so how did we come to the field? And actually this comes from the cancer field. And basically it started by work from my mentor, Georg Bornkamm, who was the first PhD student of Zuhausen, who got a Nobel Prize for HPV discoveries and so forth. And he was working on a very fast-growing tumor, which is known as Burkitt's lymphoma, which is a c-MYC-driven tumor. It's usually caused by translocations of two chromosomes, by fusion of two chromosomes, which brings the IgA immunoglobulin heavy chain under control, or MYC under control of the IgH, and thereby causing this kind of disease. It's a very super rapidly growing tumor, as you can see from the pictures here, and but it's also well treatable, luckily. So you need irradiation or some chemotherapeutics. You can almost cure the patients. But these cells, when you isolate these cells from these tumors, and this was actually work by my mentor at the time, and you can isolate these cells. Sometimes you're happy, you can establish cell lines, and but you have to do one thing. You have to really, really keep them at very, very high cell densities. They really like to hug, they like to sit together. So if you split them a bit too strongly over the weekend, it could happen that if you go below 1 million cells per milliliter, they would rapidly die. Within one day, they're all dead. And of course, he was very much intrigued by this phenotype to understand what's behind these things. And he was looking for factors and also for genes which can protect this kind of dying by seeding the cell at low cell density. And he found that actually was looking for a gene where the activity was known, but not the genes were known. It was a so-called cysteine glutamate antiporter, which is the only one. There are two transporters, but that's the main amino acid transporter which brings in the oxidized form of cysteine, it's a dyed cysteine, into the cells and at the expense of glutamate. And the activity was known since the 70s, was worked by Shiro Banna and Hideo Sato, some friends of mine back in Japan at Tsukuba. Now they retired, but Hideo is still in Niigata University. And so they cloned the gene eventually in 1999. So we started collaborating with them. And of course, he was also continuing to understand what's really behind this phenotype. What he found was kind of striking. So he could rescue this phenotype by adding thiol-containing antioxidants, such as beta-mercaptoethanol, abbreviated BME here. You can rescue this phenotype. Also, just adding nanomolar concentration of sodium selenite is sufficient to rescue this phenotype. In addition, you can also use calculators to rescue this phenotype. And so he was quite interested. It was clear it must be related to aberrant redox regulation. And so that's why he also used the system to try to clone the gene, but as I said, he failed. But instead, he found another gene, which is here depicted as GPX4. And I told the story yesterday. I'm very proud of this paper because you see here, it's published in 2001. And you see my name is right in the middle, so that's what you want. And it's published in a journal no one knows usually, unless you work in selenium research or not. A very low impact factor and almost never cited. So that's how it started. And basically, this was my topic to start a PhD, to make knockout mice for this. And we already suspected maybe the enzyme is quite important. So at the time, the name was different. It was called phospholipid hydroperoxide glutathione peroxidase, PHGPX. a long name. They have many long names with me. You will see later in my talk. And but of course, it took a while, and then they changed the name because no one can spell that. Anyway, so it was renamed to GPX4 because it was the second glutathione peroxidase 4 to be discovered after glutathione peroxidase 1. In total, we have 8 in humans, and but nonetheless, I continued working on this, and it took a long time. I started my PhD, if you see now from the date. I started in 1998, and it took us. We only published a story in 2008, so it took us 10 years to understand a little bit of the mechanism. So we found that when you knock out the gene in cells and mice, it causes a yet unrecognized form of cell death. Don't use novel, don't use new. When you publish in certain journals, they don't like that because they expect everything to be new. That's why you cannot use these fancy words. Anyway, so we knew that was a very important pathway. Also, we knew it because we made a conditional knockout at a time already, and we found that when you knock it out in neurons, these mice are born. However, they display very strong ataxia and seizures, and you have to sacrifice these animals usually around day 8 to day 10 after birth. And so we knew it's not only a vitro cell culture artifact, but also maybe very important for in vivo. We dig more into the mechanism, and we thought that maybe some lipoxygenase plays a very important role as shown here, but this was largely based on inhibitors, which later turned out to be antioxidants. So because when we made a double knockout of GPX4 and that enzyme, there was no rescue at all. And also others have now repeated this in T cells, embryonic development, and so forth. So we could cross it out, you see. There's another molecule which is in the title, which is called IFM1, or apoptosis-inducing factor mitochondria 1. We thought, we used guide RNAs at the time. I got these from a collaborator, and they said, oh, you should use these guys, they work. I said, what does that mean? So we used them, it worked a little bit. We got a kind of a rescue. So, but then when there was the advent of CRISPR, we made a CRISPR deletion in our knockout cells. And what happens? The cells still happily die. You see, we had to cross out all these things from the title. There's not much left, right, in the title, except GPX4 and oxidative stress. Nonetheless, we can correct ourselves. But it was a very nice tool as well, these mice, because we generate a cell line, which many people nowadays use. We call them PFA1 cells. A stupid name, we can discuss this later. It's an inducible knockout cell line, where basically we have 2 floxed alleles, as shown here, and we transfected them with an inducible Cre recombinase. So you simply add tamoxifen, and then this Cre would go into the nucleus, destroy the gene, you get a functional genetic knockout. At the times, there were no inhibitors. They were mostly developed by Brent Stockwell already during the same time. We didn't know each other for many, many years. But nonetheless, our system did work quite well. And as you see here from the movie, if it works, hopefully, yes, it does. You can see here, these are 2 friends or a couple, whatever you want to see. These are friends who the knockout have been induced, and it takes a while. So basically, they're still hugging and are happy and so on. Then they become a bit sour, they divorce, and all of a sudden, they die. You see this huge bubbling coming out. It's very characteristic of ferroptosis. Of course, it was a very beautiful system to be used beginning of 2011 or so. When I came back from industry, I have a little detour, where we started screening a small, around 40,000 drug-like compounds. We found around 400 inhibitors of this pathway, and one of them was a spiroquinoxaline, which we termed lipoxystatin 1, because it was very, very effective. It works in a low nanomolar range. It was stable enough to be used in vivo. This was very important for us. And so we used that to characterize the pathway much more in detail. And also this was the basis to make these so-called second-generation lipoxystatins, where we have almost completed now preclinical development with a lot of money from other donations from all over Germany, and it was a long way because we had to acquire a lot of money for doing this. It's all GMP, GLP style grade work, and hopefully we can enter clinical trials next year for the treatment of organ transplantation, especially those who require liver, otherwise they would die. We were fortunate enough to bring this molecule into the brain. This was pure serendipity. So we found a modification which brings this molecule very effectively across the blood-brain barrier. So you inject them into the belly or in the IV in a mouse, and within half an hour, everything is soaked up in the brain. So it's very remarkable. We don't know the mechanism, don't ask me. It does work. We can do it for each molecule from the same family. We just hook on this modification and we bring it in. But I'm not talking about all these developments here, so I want more about the details. And of course, as I mentioned before, and as Scott mentioned as well, this was also very handy to use. We did a CRISPR screen already back when the first CRISPR libraries came out, to search for genes which are protective. And we found one enzyme, consistently found one enzyme, which is known as ACSL4. It's also another horrible name, I know. It's acyl-CoA synthase long chain family member 4. What it does, it's a very simple activity. It just activates polyunsaturated fatty acids by attaching a CoA to it, and once it's activated, it can then be used for many purposes, but of course for lipid pockets, for ferroptosis, we need to bring them back into the membranes, because during ferroptosis, many membranes are being destroyed by oxidation. So this was kind of very nice to do, and we're still working, of course, on this enzyme, and many groups in the world as well. But coming back to GPX4, because it's still our main molecule we are still working on, because it's a very fascinating molecule. It's a very small enzyme. It only consists of around 170 amino acids, it's a cytosolic form. It also has another peculiarity. It's a selenoprotein. That means it contains selenium in its active site. And usually selenium, you see here, it's not a cofactor itself, but it's basically incorporated into an unusual amino acid, which is known as selenocysteine. And you see the difference between cysteine and selenocysteine is very small. It's just one atom, selenium versus sulfur. You would think that this is easy to do, but it's not at all easy to use. And that's why we only have 25 selenoproteins. And you see here from the activity, so you really need it. So the active site selenolate is basically reducing the peroxide in membranes, and thereby itself it becomes oxidized. It loses 2 electrons. And these electrons, of course, have to come somewhere, and usually it's glutathione. We heard this in the previous talks, that we need glutathione for many things, but one of the most important targets is GPX4. I also need to say that it's not only glutathione, which is a cofactor for GPX4, it can also use many other thiols, including cysteine. It can also use thiols from proteins, particularly then when there's no glutathione around, which is, for instance, happening in sperm development. We did also kind of work a long time ago on this. But to use selenium, as I said, is not easy. Because usually, it's interesting, because GPX4 is usually present in almost all organisms to varying degree or conservation, but it's only a selenoprotein in vertebrates. So you never find it in plants and fungi and so forth. It's only in vertebrates like fish, mammals, etc. You will find it as a selenoprotein. So how do you use the selenocysteine? It's kind of, as I said, very complex, because what's happening, you need a specific tRNA, which is then loaded with a selenocysteine, but it is a real step you've had, because first you have to load it with a serine. You put it on the serine. That's not the selenocysteine, of course. Then you have to phosphorylate the serine. Then you get a phosphoserine. And then with the help of another enzyme and selenophosphate, you convert this phosphorylated serine to selenocysteine. You see, you need already quite a few factors to do it. You also need selenium as selenophosphate. Very important. I come to this later in my talk. In addition, incorporation of selenocysteine into the polynascent chain is very complicated too, because it is encoded by stop codon. One of the 3 stop codons, UGA, as you learn it from school or at university. This would be normally a stop codon for ribosomes. So ribosomes would come in and think, that's the end, let's fall apart, job done. But it's not the case, because only the interaction between many factors binding to this very unusual stem loop-like structure in the mRNA of these proteins allow incorporation of selenocysteine into the nascent chain. It's a very inefficient process. If you compare this with a normal cysteine incorporation or serine incorporation, this ranks in the range of 1 to 5%. So it's a really, really slow process. That's why most of these selenoproteins are expressed as very low levels. Sometimes very hard to detect, very difficult sometimes by mass spectrometry and so on. Okay, but why do we have a selenium-containing enzyme? As I said before, it's usually present in plants, higher plants. It's almost everywhere, but it's only present as cysteine. So what we did, and so we danced the moonwalk, because selenium was named after goddess of the moon by the guy who discovered more than 200 years ago this atom. And so we danced the moonwalk. We replaced the selenium to a sulfur. At the time, there was no CRISPR. We started doing this in 2011 or so. And so we had to do it the hard way. So we had to mutate in embryonic stem cells. That's what we did. That's actually shown here. You convert the TGA to a TGZ. And this was kind of surprising. We had made a lot of discoveries here, because when usually the full knockout of GPX4 is embryonic lethal very early on in the gastrulation. However, when you now have these homozygous mice are born. They live around 2 to 3 weeks after birth, And then they develop very severe seizures, as you see from the movie. But they have to be in a mixed genetic background. It's an F1 generation, Black 6 versus 129 SV. You see here, there's a 2-week-old newborn, and you see just by take it away from the mother, all of a sudden you see this very, very severe seizures being developed. It would cramp, and then he moves on again. Okay, so and if you look now what's happening in these mice, if you look into the brain, and we found that a very specific subset of neurons, GABAergic alpha-albumin positive interneurons, are lacking or cannot develop in these mice, as you see here from a staining. This is now a staining against these interneurons, which is mostly gone, and you can see in green this is a marker of cell death. So these cells that basically die. So this is only one specific cell type which requires selenium in GPX4 at least to overcome this phenotype. It was kind of surprising. Of course, we also looked into the mechanism, what's going on the molecular level, and we found that when you replace the selenium with a sulfur, as shown here, the enzyme becomes very prone to overoxidation by peroxides, something which we expected a little bit, but was still surprising, and we could also show this happening in vivo. The picture has changed completely. So at the time, and then of course, Brent Stockwell came out in 2012 publishing Ferroptosis as a novel form of cell death. And since then, of course, as Scott already alluded to, there are so many papers. It's very hard to overlook the literature, even in the top journals. But nonetheless, we just try to summarize the key pathways which are now dominating or controlling ferroptosis. You see here on the very upper left, you see basically the glutathione pathway and the cysteine glutathione GPX4 pathway, which is still perhaps the most important pathway to protect from ferroptosis. There's another one, as Scott also mentioned, there's FSP1, ferroptosis suppressor protein 1, which we also discovered a few years ago, and which is also very powerful to protect membranes from lipid oxidation. The mechanism is totally different. It's on the level of radicals, whereas GPX4 does on the level of peroxides. This is kind of chemically a big difference. Meanwhile, also, of course, the cholesterol pathway has been involved in ferroptosis regulation, so some intermediates can also act as endogenous RTAs. And on the contrary, if you have too much PUFAs, polyunsaturated fatty acids, in the membranes, then you are very prone to undergo ferroptosis, because these are the proximate targets of lipid oxidation. We brought this little review up, or this little summary. It's like a recommendation, and I think if everyone is new to the field, I really highly recommend to use this, because I'm reading so many papers or have to do so many reviews, even for top journals, and they always make the same mistake, right? So, and then you see they use the wrong antibodies, they use the wrong cell lines, etc., etc., and we know they don't work, right? And then when they use inhibitors against enzymes or against species where we know they do not work, or they use inhibitors which we know are not stable enough to be used in vivo. So I really recommend to read this, and although it's maybe a bit hard diet, but I think it's something we really carefully put together. So the question is now, what's the physiological meaning of all these things, right? Because GPX4, as I said, is a very important enzyme. You can knock it out. It's the only glutathione peroxidase which is essential for life. And among the selenoproteins, perhaps the most important one. And so the question is, what is all the meaning? You knock out a very important enzyme, the animals die, so what? Okay, so that what people would ask me, at least in the cell death community. But there's a disease, and which is also, again, another horrible name, sorry for all these things, but spondylometaphyseal dysplasia Setagatien type is a disease which was discovered already in the 80s. And then in 2014, they discovered a genetic cause, because they found homozygous truncating mutations in GPX4 gene. And the disease itself is characterized by several, of course, a very severe disease. You find there's a progressive disorder. It's congenital. It shows many abnormalities, not only in the brain, but also in skeletal abnormalities. Most of these babies would die quite rapidly after birth due to cardiac arrhythmia, and you have to put them under convulsants to prevent these seizures. And also they have developmental defects in the CNS, and also very often they show cerebellar hypoplasia. And this boy here, Raghav, or actually his father, he contacted many people working on GPX4 a couple of years ago, because they found out after 1 year that the boy is suffering from a homozygous mutation in GPX4, which is now known as arginine 152H, where we'll talk about today a lot, because we think we can learn a lot from this mutation. And so we were kind of fortunate, because it's not, well, we are fortunate, but not the baby, of course, to find 2 more children carrying the same mutation, also in the States. So all of 3 are living in the States. And basically, you see here, the mutation is caused by this in exon 6, causing this homozygous conversion. The phenotype has been published, but one thing which hasn't been published was, so we got also access to the brain data from these animals and MRI data, longitudinal data. At least we got some data. We found, or at least the pathologist, they told us, neurologist, the pediatrician, they told us, all these babies suffer from progressive neurodegeneration, early onset neurodegeneration. Of course, that rang the bell, and we were looking into this, of course, and looking more into the mechanism, what's really causing the disease. And we were lucky enough as well, because Saneth, the father, he donated his fibroblasts, and those from himself and also his father, so we could use on these mutated cells. And that's what you basically see here. When you take these cells, put them into culture, as shown here. So in blue, we have the heterozygous, and in orange, we have the homozygous GPX4 expressing cells. When you now put them into cell culture and remove Trolox or vitamin E or lipoxystatin, any ferroptosis inhibitor, all the cells, they die within 24 hours to 100%, indicating the mutation does not sustain survival of the cells. They show the characteristic phenotype, so this bubbling up, we're getting very big. At the same time, also, when we use a dye to monitor for lipid oxidation, which is the hallmark of ferroptosis, you can nicely see that when you take wild-type cells treated with RSL3, these cells, they would die as expected. When you now take the homozygous expressing cells, remove vitamins or antioxidants or any other lipoxystatins, then the cells also display this very strong lipid oxidation, which is the hallmark, as mentioned. We digged more into the mechanism, and we found that there was no change in expression in father cells and son cells. So you see on the mRNA level, there's no change. There's no change on the protein level. But the very interesting part started then when my postdoc at the time, after forcing him for several months to do the experiment, when he measured the activity, GPX4 specific activity in these cells. It took a while to convince him. Eventually he did it, and then he was very, very happy, because he called me when I was traveling and said, "Marcus, I found something very exciting. The enzyme is fully active. Still, the cells, they're dying." So of course, it was kind of an unexpected result, and you see here, this is now measured by an old assay. We can reproduce the same with a newer assay we just established. Of course, you submit the story, and get a revision, you get a review, comments back, and then they say, "Yeah, but you only work in fibroblasts, you talk about neurons and neurodegeneration, but it's also true in neurons." I say, "Oh God, yeah, okay, right, how do you do this in a patient, right?" The patient's still alive, and okay, so we were kind of lucky, because we took these cells and could basically reprogram them into iPS cells. And from iPS cells, of course, we could now drive them into cortical cells, and that's what we did. And when we now put these cells under low vitamin E or absence of ferroptosis inhibitors, also these cells, they die very rapidly in the absence of any inhibitors. And you can also see the same on the movie shown in the next slide. If it runs, hopefully, yes, it does. So these are wild-type differentiated neurons. You see they're kind of happy. Whereas when you now take the same, the ones from the sun, and remove and wash out vitamin E or antioxidants, you see they all bubble up, and they all die quite rapidly after washing out the ferroptosis inhibitor. Of course, it's all phenomenal, kind of phenotype biology. So we dig more into the mechanism, because we had this discrepancy. We still have high expression, but we have a lot of activity as well, right? And still the cells, they die, neurons die, fibroblasts, they die, and so on. So we looked more into the mechanism. What we did, we teamed up with structure biologists, very hardcore biochemists, structure biologists, trying to understand what's behind all these things. And so they purified wild-type enzyme and the origin mutant enzyme with a mutation in the active site, because selenium cysteine is not very well expressed in bacteria, so we make a cysteine variant. But since we know the activity is okayish, we just look more for the overall structure of the protein. And when you now do these NMR studies, and you see colors again in blue and in orange now, it's almost an identical overlay between wild-type and the mutant protein, indicating there's no cross change in the structure of the protein. They did it more carefully then, and they found that when you look over of the protein itself, and they found a very small difference at this position around 130. You see there's a more flexibility in the mutant protein. Let's ring the bell for them and say, oh, there could be something going on around this position. So then we looked into the crystal structure, which was published by other groups already, and we also repeated that study. We found that around this position you have a loop. A loop is very small, only 6 or 7 amino acids, but the loop is very unusual because you have 2 amino acids. So these are hydrophobic amino acids, if you still remember from your biochemistry course, isoleucine, leucine, basically sticking out from the protein into the solvent, which doesn't make sense, right? You don't want to put something outside which is hydrophobic into the water. And so this was kind of interesting. And now if you look at the position of the arginine, it's basically below this loop. It's far away. It's still around 20 amino away. But you see it makes these 2 little hydrogen bonding with 2 adjacent amino acids in the loop, asparagine and alanine, thereby stabilizing this kind of loop. If you now look into the protein, the mutant protein, as shown here, when you have a histidine here, you don't have this hydrogen bonding stabilization anymore, so the loop becomes kind of floppy, so it doesn't become very stabilized. So what we did, of course, as a proof of principle, we took the wild-type protein and mutated these 2 amino acids into 2 hydrophilic amino acids. So of course, you just convert it into a serine. 2 serines, that's what we did. So we made a mutant where we now mutate the isoleucine and the leucine both to a serine. So we have an ILSS mutant. It's nicely expressed in the cells. And when you now express this in knockout cells, wash out vitamin E, that's actually shown here, the cells are dead again. So you cannot sustain survival of these mutant-expressing cells. They still have the arginine intact, but you just replace these 2 hydrophobic amino acids with 2 serines. They went on and did some bi-cell interaction studies with NMR. And you can nicely see from the pattern, this is the wild-type enzyme, you get this very nice interaction with bi-cells, as shown here by this drop. Whereas when you take the mutant protein, in the patient mutant, you don't see any interaction. When you do the same with the ILSS mutant, you see exactly the same. So it cannot really bind to the membrane any longer. You need this kind of hook or loop, as we call it. And if you do all the calculations, there's much more behind. You can see that in red colors, this is the interaction which basically interacts with the membranes. When you now take out this arginine, which is below this red part, you see this loop doesn't form properly anymore. It becomes very floppy. And of course, this is all structure biology. I'm a poor biologist, not even a biochemist. So I always make this kind of comparisons. I believe it's more like this, okay? So you have this kind of fin loop, which allows the protein to dive into membranes. So maybe you can also, we call it now fin loop. I think it's maybe more appropriate where you have this kind of surfer fin loop to ride across the waves, which are the membranes, evidently. Okay, we went on and made a mouse line, recapitulating basically this mutation. So where we now mutate in the mouse at the same position, the arginine to histidine, tried to get homozygous mice. Unfortunately, these mice do not survive. So we looked at day 9.5, so we never got a single surviving mouse. These mice, they die quite early in embryonic development. So we look at day 9.5, you see already they're completely resorbed. You have to go earlier to 7.5, which is the time when the gastrulation has just finished. And then you see start to be resorbed, which indicates umbilical lethality around the same stage as we make a full knockout. Okay, it was kind of unlucky because we could use this mouse, while it's kind of a little bit, frustrating if you make a mouse and you don't have any big phenotype. So what we did, we took a trick. So we crossbred these mice with a mouse which has a conditional allele, which I mentioned at the very beginning, the floxed allele. So now we make 2 mouse lines, one mouse line where we have 2 floxed alleles with a CAM-CRE-RT2, which is the driver to delete GPX4 in cortical neurons. And the other one where we have 1 floxed allele where we can inactivate the wild-type enzyme, but we keep the arginine intact. So these are the heterozygous mice. So then we can really look, we can bypass embryonic lethality and then induce the knockout when we want to, at will. And what happens in these mice when you now delete both alleles of GPX4? So these are mice which have been induced for 6 months or so, and I'll show you later some pictures. You can easily see these are the knockouts. These are the wild type. They're running around. These ones, they have a problem. You see, they become They become very tame. You can put them on the hand and you can touch them. They usually would bite, but they become quite tame. They have the only problem in feeding. You have to put the food in the cage, and they have a bit of problem in cleaning, so they're a bit greasy in the fur. So, but otherwise they're kind of okay. Okay, and I'll show you later some pictures. You won't believe it. So we looked, of course, in cell death. So then we did our tunnel staining, and now I have 3 colors. One color is always easy to remember. It's a wild type. The red one is the full GPX4 neuron-specific knockout. And then we have the orange, which has this one origin allele intact. But you see there's no difference. If you stain for tunnel, which is a mark of death, a general mark of death, in wild type, of course, and there's no change at 2 and 4 weeks upon knockout induction. Whereas in the full deletion, and also in the origin expression, you see this nice staining positivity for tunnel staining. That's a kind of a cumbersome assay, and we heard in the afternoon already it's quite difficult to stain brains for tunnel because it's a very rapid process, and also we have our hard time to establish the system. So we use now another approach where we now measure the damage in the blood of these mice. We use we stole this setup from neuropathologists. So when they use neurofilament light chain, which is a marker of neurons, which is then shedded when they die, it's shedded into the plasma of patients or of mice. Then you can basically monitor in a quantitative way. You can monitor the increase of neurofilament in the plasma of these mice. And you can see here normally in a healthy mouse there's around 50 picogram per ml, like in a human being. When you now induce, you see this massive increase, and which really peaks at 2 weeks upon knockout induction. Then it drops down again because most of the neurons are dead, and then you see some other phenotypes. And we also compared this, of course, by MRI imaging of these mice. So we looked at control mice. These are to the very left. In the middle, we have the full knockout, and to the right, we have the mutant mice. Of course, in the control, nothing happens. You see, there's a top view. These are the eyes, of course. These are cortical hemispheres, cerebellum. And you see there's no big change over time as expected. Whereas in the knockout, you see that already it's 2 weeks. We see this enlargement of the ventricles. And at 2 months, or respectively 6 months upon knockout induction, you see this massive loss of cortical mass. You see almost a similar phenotype in the origin expressing. You see this enlargement of ventricles, but we do not see this loss of cortical mass from outside. We still do not understand, maybe you have an idea why it's happening, but there's a different picture. And also when you now look at top view of these isolated brains, you can now see there's a control mouse. This is the knockout shown at 6 months. You see there's an 80% loss of cortical mass. And here you see still an intermediate phenotype indicating there must be some kind of suppressor genes which protect this tissue. Of course, we looked also in inflammation, and we stained astrocytes and the microglia cells. And now I show quite a few slides only on the GPX4 knockout. And you can nicely see that already it's around 2 weeks upon knockout induction, you have this strong increase. Or you see already at 1 week a little bit of astrogliosis with a strong stain for GFAP, which is very pronounced at 4 weeks, and microglia activation comes a little bit later at 2 to 4 weeks respectively, and then it drops down again if you look later, but astrocytes remain high over all the time we are looking at. We went together with the collaboration in Hamburg. We did some studies. We did a more granular view of the microglia, so because they changed their morphology, as you have heard today in the afternoon in some talks. You see at 2 and 4 weeks, you see this change in morphology of the microglia. And I had no time about all these things because people were teaching me all this interesting stuff. Because usually what you can do is you can also analyze these microglia using different markers. And if you have this so-called disease-associated microglia, they change the pattern of expression. And that's what we actually see when we now look at 12 days after tamoxifen induction. There's no big change between wild type and knockout. However, when you look at 4 weeks, you see this nice segregation of the different markers indicating homeostatic stage transition into a disease-associated stage. Then, as I said, you submit the paper, and then the comments come back. You think the reviews are great sometimes. And the editor says, "Oh, why don't you make another knockout?" I said, "What?" I said, "Oh, I never heard this before." So, but then she said, "Okay, but what do you want?" "Yeah, can you make a cerebellum? Because the patients have a cerebellar phenotype. You should also show this." 10 years ago, we had a small paper on this, similar, but using a different Cree driver. And I said, "Yeah, but you should use L7 Cree." I didn't know what L7 Cree, and so we said, "Okay, let's do it because you're the boss." All right, so we did it, and so we had to get animal approval in Germany, which is not easy to get. And then you have to order the mouse, you have to do the breeding, do the analysis, and geez, and you don't know what's the phenotype anyways, but it came out very well. So what we made a deletion of GPX4 in the cerebellum using LC-driven mice, 7-Cre-driven mice. And this Cre line is only, or this Cre is only active, I think, around 3 weeks after birth. So we wait around 6 weeks because then these mice show a very peculiar phenotype, movement phenotype, ataxia. And then we look at the Purkinje cells in cerebellum, you see they're mostly gone. Also, when you look for neurofilament light chain, it goes up. And if you now look at the histology, immunohistochemical stainings, you can see that basically all calvin is a marker of Purkinje cells, they're all gone. At the same time, we have this massive astrogliosis, Bergman astrogliosis. And so the editor was very happy to see that. So we also. Anyway, so we took these mice, not these ones, but the other ones, and of course tried to treat them with lipoxystatin. I just showed the data on lipoxystatin because we know lipoxystatin itself can cross the blood-brain barrier, but very slowly, and you need more than the new ones. But nonetheless, we took LIP1 at different concentrations and at 30 mg per kg. You can see that at least you can suppress this kind of neurodegeneration. You will never rescue, to be honest. If you look later, you can never 100% rescue the phenotype. You get a delay, a strong delay, but the neurons will still die. It's just simply too brutal. We teamed up with Matthias Mann and Ashok at the Max Planck in Munich because we sampled from these animals sufficient material to do a label-free proteomics analysis, see more what's going on behind this. And they did a very nice study. It was all blinded, double blinded, and they came back with fantastic results, except 2 samples. Totally the opposite. I said, oh, what is this again? Right? I said, do you still have a little bit piece left which we can use? They said, yes, we have it. And so we took it and made a Cre PCR, and there was a wrong genotyping. It was a mixed up. So one was Cre positive, so it was a real blind experiment. So everything looked perfect. So you see many proteins in red are upregulated, especially 2 and especially in particular at 4 weeks upon knockout induction, whereas many proteins are lost in these samples. And of course, if you now do some kind of PCA analysis, you see they nicely cluster the knockouts at 1, 2, and 4, whereas the controls they remain together. We did some KEC analysis, and then my bioinformatician came and said, Marcus, we find so many Alzheimer's disease genes. I said, what? That doesn't make sense. I said, right, we don't touch any Alzheimer's genes. But nonetheless, so we found, if you do the KEC analysis, that Alzheimer's disease at 2 weeks is already kind of comes up, which becomes very prominent at 4 weeks upon knockout induction, which was really surprising, even for me. I had no clue what we're doing, quite naive. And so we took that data, and also with the help of Scott, basically we were looking more into comparisons between human data sets, proteomic data sets, and our data sets. And if you look at 1 week, we don't see any big overlap between human, but at 2 weeks, we get already substantial overlap between human in blue and in yellow, the mouse data sets, which becomes even more pronounced at 4 weeks upon knockout induction. If you now look at individual genes, in these data sets, you find many genes which have been already associated with Alzheimer's disease, like APP, clusterin, which is ApoJ, ApoE, and many more. So it was very surprising to find all these hits. Of course, that needs to be worked out now, but there's a lot of work. But we found another gene which intrigued me for quite a few years, which is known as PRX6, peroxidoxin 6. It's another redox enzyme. And so we thought, could this be a compensatory mechanism, upregulation due to loss of GPX4, or in Alzheimer's disease, experience high oxidative stress? So what is PRX6 doing? It's 1 out of 6 peroxidoxins, and it was discovered by Aaron Fisher a long time ago. And he proposed that this enzyme has phospholipid hydroperoxidase function. It's a non-selenium-containing enzyme, it's an unusual peroxidoxin, 1 out of 6 I mentioned, has 3 different active sites, or at least 2, has a phospholipase A2 activity, has a peroxidase activity on cysteine 47, and another alternative cysteine, whatever that means. And so what was reported, not by us, but by other people, that this cysteine 47 can reduce peroxides, phospholipid peroxides to alcohol, akin to GPX4. So we wondered, it's very logical, why you knock out GPX4, maybe cells that try to compensate, upregulate PRX6. So what we did, very simple, we took the enzyme, overexpressed in our knockout cells, I showed in the movie before, and then you see what's happening, it happens nothing. Okay, the full knockout would die, these are death cells. When you now overexpress PRX6, you see there's no rescue at all. Whereas when you take FSP1, which is the strong backup, you get full rescue, or another enzyme which is important for biosynthesis, discovered by others, you can also completely rescue this phenotype. Kind of frustrating, right? It's up in Alzheimer's disease patients in our data sets. Then I told Junior, I said, then knock it down, right? Why not just knock it down for fun? He did it. It was very interesting. So when you knock down PRX6 in fibrosarcoma cell lines, you can do it in many cells, it doesn't matter. What you see here is a very nice knockdown, but you also see that GPX4 is going down very strongly. And this was kind of very surprising. And this was also very specific. So when you now knock out basically all other peroxidoxins like 1, 2, 3, 4, and 5, you knock them down, you see there's no change at all on GPX4. It's really specific to this one enzyme. And when we take these cells now and put them on the low selenium medium, which you can do, especially formulated. So to the left you have the wild type, to the right you have the knockouts. You see the wild type, they don't care. If you put them in a no selenium medium, whereas the knockouts, they all die again due to ferroptosis. You see this massive bubbling up. And to summarize a long story, otherwise it would need a lot of time here, we basically identified PRX6 as the missing selenium carrier inside the cell, which transports selenite coming from either the inorganic way or the organic way, being loaded on this cysteine 47. I didn't show data, but believe me. And then it delivers selenium to selenophosphate synthetase, which is then phosphorylated, which is then important for the last step of the tRNA synthesis to make selenoproteins, including GPX4. That's not the end of the story. And we got scooped, so I had to bring out the story. So it was a Japanese group from Kyoto. It published in June last year. And my previous co-worker, we also discovered his work on the same, so we had to put everything together quite rapidly, so we couldn't do all the in vivo work because it takes too long, evidently. But nonetheless, we got the mice already, the PRX6 knockout mice, to see whether we can also reproduce this phenotype, what we see in cell culture, also in vivo. And when we did the analysis, we looked in PRX6 knockout mice in liver or in kidney. So these are real knockout mice, believe me. You see there's no change in GPX1, which is another selenoprotein, or in GPX4, as you see here. But the big, nice part came when we looked into the brain. In the brain, you see the same what we see in cell culture. So GPX1, and in particular GPX4, is going down. So indicating there must be some kind of regulation, which we still don't understand. We'll work on this, because PRX6 is up in Alzheimer's disease, maybe as a stress signal. It's also up in GPX4 because they think they can make more GPX4, but they can't because GPX4 is gone in our model. Of course, not in Alzheimer's patients. They would not survive. So there must be some kind of regulatory circuit, which is important. And we believe that this maybe underlies some of these diseases. And with this, I come to my penultimate slide, which is a nice summary, I think. I like to make all these old links because ferroptosis has been described in 2012, but there's a lot of research going on since the 30s of last century. And there was even one earlier paper published by Alois Alzheimer. Everyone should know him. He made a paper in 1911, and he made a very nice statement. And this is all summarized here. I think it was translated because at that time many people published in German. And the last sentence is the most important one, which I really like, because he said the plagues, the A-beta plagues, are not the cause of senile dementia, but only an accompanying feature of senile involution of the CNS. And if you now look at all the drugs which have been approved in Germany, in America, and all over the world, right, do you still believe that A-beta is such a very important trigger of death? I'm not quite sure. I think that we can use this for discussion for the afternoon, for the evening. And with this, I hope that at least we can contribute with manipulating ferroptosis in one or the other hand, that we can at least treat some of the disease or at least understand the mechanism of death of the neurons and other cells, such as, of course, in ALS and Alzheimer's disease dementia, perhaps in Parkinson's disease, perhaps in Huntington's disease, certainly in multiple sclerosis. At least the models are quite, when you use inhibitors against ferroptosis, you can nicely suppress the degenerative part in MS models. During ischemic perfusion conditions, like in ischemic stroke, ischemic myocardial infarction, organ transplantation, we can always get very nice protective activity when we use the compounds. On the other hand, of course, using this form of cell to trigger certain, I don't have time to talk about it, there will be tomorrow the chance, So to hope that we can kill certain types of cancers has been shown now that certain tumors are really dependent. There's a new paper coming out, or two new papers. It's important for metastasis. So during metastasis, cells become super vulnerable to ferroptosis. So what I heard from the metastasis field is half of the groups are working on ferroptosis now to use that kind of Achilles heel. And of course, maybe as a standalone therapy, or maybe in combination, likely in combination with immune checkpoint blockade. And with this, I come to very important last slide. These are the people involved. I have a wonderful team, especially Aika Mishima. I didn't talk so much about him, but he made some very great breakthrough discoveries. Bettina, she's my right hand. She's doing all the development. Toshitaka Nakamura was also involved in FSP1 characterization. Svenja Lawrence, she's here. She did all the work together with Adam and others on the brain phenotype. I have a very strong team. There are many, many more. These are the people doing the structure biology, and I have many, many collaborations worldwide. And if you're interested in ferroptosis, I hope so. At least some of you, at least will be hopefully the future. Please come next year. We have a wonderful meeting again. Not in China, it will be in Japan. So in November. So if you like Japanese culture, it's close to Kobe. It's a beautiful island next to Kobe. Kobe steak and other things. So please come and join and register. And thanks for listening. And I'm very happy to be here. And thanks again.
Thank you very much, Marcus. That was a fantastic tour de force of what is an extraordinary amount of work in the space of a relatively small period of time. You really have defined the field, and we all have a sense of the power of curiosity in your research, and certainly that was something articulated in the talk last night, that a lot of these discoveries have come from experiments that you did in your PhD and sort of odd findings that didn't make sense at the time were stuck in the freezer but never left your conscience or consciousness and have led to major advances in the field. So thank you very much for sharing that with us tonight. Marcus has very kindly agreed to take some questions. So Scott, I wouldn't mind if you would take the mic around. I think there's one just there. Do we have any questions? You geesh, always quick out of the gate.
Confusion. Marcus, amazing talk. Given the role of selenium in cell death, have you? I mean, I'm sure you thought of this, but selenium as an IV therapy in conditions that are earmarked by ischemia, reperfusion injury. I'm thinking like targeted interventions that is possible after acute myocardial infarction, during clot removal, or even stroke. Have you considered giving selenium as a therapy to show any benefits?
I think the selenium field has become quite quiet last 20 years or so because of the failed trial of the SELECT trial, which was a selenium vitamin E supplementation trial, where they discovered there's no effect if you supplement, right? And this is just on a longer time scale. As a treatment, people of course think about it. The problem is only you need always, when you want to add selenium or selenite, which can go across the blood-brain barrier, it will take a while to make the proteins, make protective. So acute settings will be very difficult, right? In addition, if you want to treat Alzheimer's disease, it has been tried, it doesn't help. Because I think once you have an impairment in the entire axis to utilize selenium in its real form, natural form, maybe that's a main problem. Because what I showed today, it is not everything as easy as I showed it today. Because usually in nature, normally in a human being, you ingest your source of selenium. Usually it can be fish or any dietary components. You have to bring it to the liver, and the liver will be incorporated in another selenoprotein, which is known as selenoprotein P, which contains up to 10 selenocysteine residues. That one will travel across the bloodstream, goes to the blood-brain barrier, will then bind to another receptor, which is the ApoE receptor 2, also known as LRP8. And only by this mechanism can be taken up behind the blood-brain barrier. They need to be degraded again and incorporated again and make again form a tRNA. And then this mechanism after the blood-brain barrier is not really clear how that works. You see, if there's any impairment in the entire axis, you have a problem, right? And it's a slow process. So I think for acute settings, treating with selenium might be too late because you need to make the enzyme. It takes a while. It's a very inefficient process. That's why, at least for acute setting, I would take always fructose inhibitors if possible, like for ischemic stroke, myocardial infarction, and others. I think that's the better way to do. Long run, maybe it helps, maybe, yeah. But this needs to be done if there's no impairment in the axis, okay? There's so many proteins involved, there's really a major problem. Because in nature, there's normally no sodium selenite in water or other source. We always take it up in protein form or as selenomethionine. That's the main caveat, okay?
Really fascinating work. I was particularly sort of taken by the strong effect in your mouse model where you've got this early embryonic lethality. What's happening in the patient who is also homozygous that they're surviving?
Yeah, if you would know this, I would tell you maybe. But no, the thing is, of course, it was kind of disappointing to get the phenotype because we knew this phenotype from the full knockout, right? It looks exactly like the same. It was like, this cannot be. The patient survives, right? But of course, the patient is outbred. Usually, these mice are inbred, so it was made on a black 6 background. So what we did the same trick in the one movie I showed, these very severe seizures. This was a mixed phenotype, a mixed background, F1, where we have a half black 6, half under 29 SV. So we did exactly the same strategy for this mouse line. For the arginine-expressing mice, we did the same F1, interbreeding again, and then search for survivors. We didn't get any, unfortunately. So it's even more severe than the other one, the cysteine variant. We did some additional studies. Now we can use these iPS-derived organoids. We can do some other studies, and also we can do early-stage mesoendoderm development, and we do see some kind of compensation, but we do not know what are the genes. So that's what it's ongoing now. So we're now looking for suppressor genes, which are germline specific, which is pretty cool, but we don't know the identity of this. We are doing CRISPR screens and so on in these iPS-derived systems. I want long sort of beer. Oh, maybe not. Some more questions?
One more question.
No, wait, okay.
Thanks, very nice talk. I'm wondering, since ferroptosis is so important in many diseases, are there any clinical trial, particularly in neurodegenerative disease, based on ferroptosis theory?
I think not that I know. I know the field is very active. I don't know what industry is doing so far. They're usually very quiet until they publish the clinical trial, right? So I just know that at least for inhibition, there's nothing ongoing. Because the problem, a little bit the problem is, well, industry is very conservative, right, as we know, because they always want to have a target. Okay, number one, you need a target, a real target. So basically a protein target, that's the only thing they accept, okay, usually. It's very difficult to find a protein target whose inhibition could ameliorate a phenotype. Of course, we've discovered ACSL4, one of these fatty acid ligases, a long time ago, and you get very strong rescuing effects in cell culture. If you knock out ACSL4, cells become super resistant to ferroptosis, which implies that the knockout, it would be a nice target, and I know industry is working on this. We have the mouse, imagine, we have the mouse since 2016, and we never published on this. Why? Because we don't have big rescues, right? And still, industry is working on this. So what industry doesn't like to come back, because I still believe radical traps, these RTAs, they're all radical trapping antioxidants, which have no target. They have a target, which is the membrane, which is a boring target. It's like inhibitors for DNA, which they use for treating cancer, right? There's also no target DNA, right? So anyway, so these are some wild comments. But anyway, so that's what they don't like. They don't like radical traps, okay? Usually because they're redox active. For them, they're hard to control, you know, and so target engagement they don't do and so on. Biomarkers are still missing. That's another caveat. So I think it will be difficult, but believe me or not, at least for acute settings, this is the way forward. And we can control. We're not stupid. We know what we have to look for, which interaction, which hERG assays, we have to do which receptor interaction, we need to do all these, of course, toxicology assays. That's what we're doing. So there will be a path forward, but I think it has to come from academia, because industry is too conservative, because there's no real protein target so far. That's one part. For inhibition. For activation, it looks different. There are targets like GPX4 is difficult, it's a very difficult target. XCT is a beautiful target. It's very hard to target because it's a transporter. FSP1 is a beautiful target if you find a tumor which is dependent on this. And so basically, because GPX4 might be too lethal, but I know that industry is strongly working on this. At least on XCT, I know 3 companies, one in Japan, one in Belgium, one in France. They are basically large pharma. They're working, they have inhibitors, and they will go into the clinics in 2 years or so. So it's just a matter of time. It's not a matter if, it will come. Yeah, and that would be even bigger, I think, the field. You saw the first slide from Scott, so it would be probably steeper even. I don't know. Okay, talk too much. Okay.
Please join me in thanking Marcus for a fantastic seminar. And before we close, I'd like to invite Florey board member, the Honourable Gabrielle Upton, to come forward and give Marcus a small gift of our appreciation.
On behalf of Florey, thank you very much for visiting us, taking time, educating the students, and giving me know very little about neuroscience. Congratulations.
Thank you so much. Yeah.
Don't drink it all tonight.
Thank you.
Scott.
Thanks, Gabrielle. Thanks, Marcus. And reiterated thanks to Allan and Maria Meyer, whose generosity has made tonight possible. We'll now recline in the foyer or retire to the foyer for some refreshments and plenty of opportunity to speak with Marcus. So thanks again for coming.